Somewhere inside your body, right now, there is a protein behaving like a quantum computer. Not in a lab freezer, not near absolute zero. Inside warm, wet, chaotic living tissue, the exact environment scientists have spent 100 years telling us would destroy quantum behavior instantly. And a team at the University of Chicago just proved that rule wrong. For a full century, physicists lived by one unbreakable law. If you wanted to see quantum effects, you had to freeze your system down to temperatures colder than deep space. You had to isolate it from light, from vibration, from every stray magnetic field. Quantum states were treated like glass. One wrong touch, and they shattered. That is why quantum computers today look the way they do. Google's machine sits in a giant gold chandelier of wires, cooled to a fraction of a degree above absolute zero. IBM's systems need entire rooms just for refrigeration. These machines are unbelievably powerful, but they are also unbelievably fragile. They cannot survive room temperature. They definitely cannot survive inside a living cell. Then in August 2025, a team led by David Aushalom and Peter Maurer at the University of Chicago's Pritzker School of Molecular Engineering did something nobody thought was possible. They took a glowing protein, the same kind of protein that makes jellyfish light up green, and turned it into a working quantum bit, a qubit, the basic building block of quantum technology. And it worked at room temperature inside living mammalian cells and bacteria. Let that sink in for a second. Not a machine built in a clean room, not a chip etched in silicon, a protein, something your own cells could theoretically manufacture on their own, functioning as a real, measurable quantum object, or while sitting inside a living, breathing biological system. The paper was published in the journal Nature, and it did not go unnoticed. Physics World named it one of the top 10 scientific breakthroughs of the entire year. Scientists across the world who had spent decades trying to keep quantum systems away from warmth and noise suddenly had to ask themselves a very uncomfortable question. What if biology had already solved the problem we've been struggling with for 100 years? Before we go further, hit that like button. It genuinely helps this channel keep making videos that dig into the science most people never hear about. To really appreciate why this matters, you need to understand why cold was ever the requirement in the first place. Heat, at the atomic level, is basically random motion. Atoms jiggle, vibrate, and collide constantly. The constant motion is called thermal noise, and it is the natural enemy of anything quantum. A fragile quantum state can get knocked out of its careful configuration by even the tiniest bit of that random jostling. That's why superconducting qubits, the kind used by companies like Google and IBM, are cooled to temperatures colder than outer space, close to absolute zero, inside massive refrigeration systems called dilution refrigerators. At that temperature, atomic motion nearly stops, and the quantum state can survive long enough to actually be useful. It's an incredible engineering achievement, but it's also expensive, bulky, and completely impractical for anything involving living tissue. Other approaches exist too. Companies like IonQ trap individual charged atoms using lasers and electric fields, another method that requires extreme precision and isolation. Diamond-based quantum sensors, built from tiny defects inside a diamond crystal lattice, can actually work at room temperature, which is part of why they've become the gold standard for quantum sensing outside of full computing applications. But even diamond sensors are external tools. You still have to bring them to whatever you're measuring. And getting a diamond crystal inside a living, functioning cell without destroying it is essentially impossible. This is exactly the gap the University of Chicago team was trying to close. They wanted a quantum sensor that didn't need to be brought to biology. They wanted one that biology could build for itself from the inside out. So how do you actually turn a protein into a qubit? To understand that, we need to quickly understand what a qubit even is. A normal computer bit is either a 0 or a 1. Simple, binary, predictable. A qubit is different. It can exist as a 0, a 1, or some strange and between combination of both at the same time. This is called superposition, and it's the reason quantum computers can, in theory, process certain problems exponentially faster than any regular computer ever could. But qubits also have something called spin. Think of spin as a tiny internal compass needle pointing in a particular quantum direction. That spin state is incredibly sensitive to its surroundings. Magnetic fields, electric fields, even tiny vibrations can nudge it. Normally, that sensitivity is the enemy. It's what causes qubits to lose their quantum information, a process engineers call decoherence. It's the reason quantum computers need to be so isolated in the first place. Here's where the University of Chicago team flipped the entire logic on its head. Instead of treating sensitivity as a flaw to be eliminated, they treated it as a feature to be exploited. If a quantum system is naturally sensitive to its environment, why not use that sensitivity to measure the environment itself? The protein they chose is called enhanced yellow fluorescent protein, or EYFP. It belongs to the same family of glowing proteins originally discovered in jellyfish, the ones that gave us green fluorescent protein, a discovery so important it won its original researchers a Nobel Prize. These fluorescent proteins already had one unusual trick. They glow when hit with light, but nobody had confirmed they could also hold and reveal quantum spin information, at least not in a way anyone could reliably use. Avshalom and Maurer's teams found a way to excite this protein with light and then read out its spin state optically. In simpler terms, they found a way to ask the protein a quantum question and get a quantum answer back, just by shining light on it and watching how it responded. And the protein answered consistently, reliably, inside actual living cells. It's worth appreciating just how significant the fluorescent protein family itself has already been to science. The discovery and development of green fluorescent protein, the original member of this glowing protein family found in jellyfish, was considered so important that it earned the 2008 Nobel Prize in chemistry. That discovery alone transformed modern biology, giving researchers a way to literally watch genes turn on and off and see proteins move inside living cells in real time. What the University of Chicago team has done is take that already legendary biological tool and hand it an entirely new job. Not just glowing to reveal where a protein is located, but glowing in a way that reveals its actual quantum spin state. Same family of protein, same basic biological trick of using light, but repurposed for a job nobody originally designed it for. If you're picturing superposition and spin and struggling to visualize it, here's a simple way to think about it. Imagine a spinning coin. While it's spinning in the air, it isn't clearly heads or tails, it's some blurred combination of both possibilities at once. Only when you catch it and look does it settle into one definite state. Quantum superposition works a little like that spinning coin, except the coin can be influenced by things like magnetic fields around it while it's still spinning, and reading that influence tells you something real about its surroundings. This matters enormously because of where these qubits can be placed. Traditional quantum sensors, the kind built from defects in diamond crystals, are incredibly precise, but they are also external tools. You have to bring the diamond sensor to whatever you're trying to measure. You can't easily get it inside a living, functioning cell without disrupting everything around it. Protein qubits are different because they're made of, protein. They can be genetically encoded directly into the DNA of a living organism. That means the cell itself builds the qubit. It manufactures its own quantum sensor, positions it exactly where the researchers want it, down to the atomic scale, and keeps producing more of them as it grows and divides. Researchers involved in this work said something remarkable about the precision involved. Biology doesn't just make one or two identical copies of something. It makes trillions upon trillions of identical copies, each one positioned with atomic level accuracy, something engineers building quantum devices in a lab could never realistically achieve using current fabrication technology. Think about what that means for scalability. One of the single biggest obstacles in quantum computing right now is manufacturing consistency. Every physical qubit built in a lab is slightly different from the next, and those tiny differences cause errors. Biology, through billions of years of evolution, already solved this exact problem, just for a completely different purpose. Now scientists are asking whether they can borrow that solution, and the sensitivity of these protein qubits isn't just a neat trick. According to the research team, these biological sensors can detect signals thousands of times stronger than existing quantum sensors currently in use. That's not a small improvement. That is a potential leap in how precisely we can measure what's happening inside a single living cell. So what do you actually do with a quantum sensor that lives inside a cell? The applications researchers are already discussing are honestly a little mind-bending. One of the biggest is something called quantum-enabled nanoscale MRI. You already know what an MRI machine does. It uses magnetic fields to create images of the inside of your body, usually to look for injuries, tumors, or abnormalities. Now imagine an MRI, but instead of scanning your entire body, it can map the inside of a single cell, atom by atom. That's the scale researchers are talking about, not organs, not tissues. Individual molecules, individual proteins, individual points inside a cell, mapped with a level of resolution that current medical imaging simply cannot reach. Peter Moore, one of the lead researchers, explained the concept in a University of Chicago podcast interview. He described how, when engineers build a traditional quantum computer, they go to enormous lengths to shield the qubit from every possible outside influence. Magnetic fields, electric fields, temperature changes, physical forces, all of it. With this new approach, they essentially reversed that goal entirely. Instead of shielding the qubit from a specific signal, they engineered it to become mainly sensitive to the exact signal they want to measure. That reversal is the whole philosophical shift behind this breakthrough. Traditional quantum computing says isolate everything. This new field, sometimes called quantum biology or quantum sensing, says instead tune the qubit to listen for one very specific thing and let it live inside the noisy chaos of biology, because that specific signal will still come through clearly. What kind of signals are we talking about detecting? Researchers mention things like the magnetic and electric fields surrounding a target protein, sometimes called its hydration sphere, essentially the arrangement of water molecules and ions clustered around it. That arrangement changes depending on what the protein is doing, whether it's folding correctly, misfolding, interacting with another molecule, or malfunctioning in ways associated with disease. This is where things get personal for a lot of people watching this. Misfolded proteins are directly linked to some of the most devastating diseases we know. Alzheimer's disease involves the misfolding and clumping of a protein called amyloid beta. Parkinson's disease involves misfolded alpha-synuclein. Many cancers involve proteins that fold incorrectly or function abnormally at a molecular level, long before any tumor becomes visible on a traditional scan. If scientists can eventually use protein qubits to watch these folding processes happen in real time, at the atomic scale, inside living cells, it could completely change how early we catch these diseases. Instead of waiting for symptoms or waiting for a tumor to grow, large enough to see, doctors could potentially detect the earliest molecular warning signs, years before anything else would reveal them. It's worth pointing out that early detection like this isn't a small deal. Cancer treatment outcomes, in particular, often come down, almost entirely to timing. A cancer caught in its earliest molecular stages, before it has formed a visible tumor, is generally far more treatable than one caught after it has already spread. If quantum sensors can eventually flag the very first molecular signs of malfunction inside cells, the entire timeline of diagnosis and treatment could shift dramatically earlier than what's possible today. The same logic applies to neurodegenerative disease. By the time a patient shows visible symptoms of Alzheimer's or Parkinson's, the underlying molecular damage has often been building silently for years, sometimes decades. A tool capable of detecting the earliest protein misfolding events long before symptoms appear could completely change how these diseases are studied, tracked, and potentially treated in the future. To be clear, we are not there yet. The researchers themselves have been careful to say that these protein-based qubits do not yet match the sensitivity of the best diamond-based quantum sensors currently used in labs. This is a first-generation breakthrough, not a finished medical tool sitting in hospitals tomorrow. But the pathway it opens is enormous, and in science, opening an entirely new pathway is often more important than any single result along it. The National Science Foundation clearly agrees because it has already stepped in with additional funding specifically aimed at pushing this technology forward. A faculty early career development program award is being used to tackle two very specific technical challenges holding the field back right now, something called photobleaching and something called readout fidelity. Photobleaching is a problem familiar to anyone who has worked with fluorescent proteins before. Basically, when you keep hitting a glowing protein with light over and over, eventually it stops glowing as brightly. It fades. For a quantum sensor that depends on being read optically again and again, that fading is a serious limitation. Solve photobleaching, and you extend how long and how reliably these protein qubits can be used to take measurements. Readout fidelity is the second challenge, and it refers to how accurately researchers can read the actual quantum state of the protein. If your readout isn't precise, your measurements aren't trustworthy, no matter how sensitive your qubit is underneath. Improving readout fidelity means getting cleaner, sharper, more dependable quantum signals out of a living, moving, chemically busy cell. Both of these challenges are being addressed with the long-term goal of reaching what researchers call the single molecule level. That means being able to track the quantum behavior of one individual molecule inside one individual cell with confidence. If that goal is achieved, biologists would gain a tool that lets them study the fundamental machinery of life at a resolution nobody has ever had access to before. It's also worth being honest about how science actually works at this stage. One breakthrough paper, even one published in a journal as respected as Nature, is not the final word. Real scientific progress depends on other independent labs, successfully repeating a result, refining it, and pressure testing it from every possible angle. That process of replication is already underway across the quantum research community, and it's exactly what will determine how quickly this technology matures from an exciting laboratory result into something with practical real-world applications. That kind of scrutiny isn't a weakness of science, it's the entire point of it. Extraordinary claims and turning a living? Protein into a quantum bit is about as extraordinary as claims get, require an extraordinary amount of verification before the wider scientific community fully accepts them. The fact that this result has already been published in Nature and recognized by physics world as a top breakthrough suggests it has cleared a very high bar, but the real test will be how it holds up over the next several years of follow-up research. Well, this entire research effort sits inside something called the Berggren Center for Quantum Biology and Medicine at the University of Chicago, a program specifically built around the collision of two fields that used to have almost nothing to do with each other, quantum physics and biology. For most of scientific history, these were separate departments, separate journals, separate conferences, separate ways of thinking entirely. That separation is starting to collapse. And honestly, it makes sense when you think about it for more than a few seconds. Quantum mechanics is the actual foundation of reality at the smallest scales. Chemistry runs on quantum rules. Biology runs on chemistry. So, in a very real sense, quantum effects were always happening inside your cells, whether or not scientists had the tools to detect them. Interestingly, this isn't even a brand new question for science. Back in 1944, the physicist Erwin Schrödinger, famous for his work on quantum mechanics, gave a series of lectures later published as a short book called What is Life? In it, he asked whether the strange rules of physics he helped discover might be needed to fully explain how living organisms work. His lectures went on to influence an entire generation of scientists who eventually helped found the field of molecular biology. Schrödinger never had the tools to test his ideas directly, and for decades, the question of whether quantum effects play a meaningful functional role inside biology remained mostly theoretical, more philosophy than laboratory science. What the University of Chicago team has done is take a version of that 80-year-old question and, for the first time, answer part of it with hard, reproducible, peer-reviewed experimental data. What changed is not the existence of quantum behavior inside biology. What changed is our ability to finally see it, measure it, and eventually use it. For 100 years, we assumed biology was simply too warm, too wet, and too chaotic for anything quantum to survive inside it. This breakthrough is one of the clearest pieces of evidence yet that assumption was wrong. David Aushalom, who also directs the Chicago Quantum Exchange, has spent much of his career working on more traditional quantum systems, the kind built from engineered materials in highly controlled lab environments. His willingness to pivot toward biological qubits says a lot about where serious researchers now believe the most exciting frontier actually is. It's worth pausing here to be precise about language, because headlines love to stretch the truth. This is not, technically, a full quantum computer sitting inside a living cell, capable of running complex algorithms the way a machine from Google or IBM can. What researchers created is a single functioning qubit, a quantum sensor, the fundamental building block that quantum computers and quantum sensing devices are made from. But that distinction, while technically important, doesn't make this any less extraordinary. Every quantum computer ever built started with the challenge of creating one working, reliable qubit first. This breakthrough proves, for the first time, that a living biological system can produce and sustain one entirely on its own, using its own natural machinery, without a dilution refrigerator, without a vacuum chamber, without any of the extreme engineering quantum computers normally require. If you're enjoying this deep dive, go ahead and subscribe. This channel breaks down the science stories that sound like science fiction but are completely, verifiably real. Let's talk about what the future of this research could realistically look like. Researchers involved in the project have discussed the possibility of using biological self-assembly, meaning the natural way cells organize and build structures, to eventually create organized arrays of these protein qubits. Imagine not just one glowing protein, qubit inside a cell, but structured networks of them, communicating and cooperating, essentially built by biology's own molecular machinery rather than human hands. That idea directly attacks one of quantum computing's oldest and hardest problems, scalability. Right now, adding more qubits to a traditional quantum computer means more engineering complexity, more cooling requirements, more room for error. If biology can grow and organize large numbers of qubits on its own, following the same self-assembly rules that build every cell in your body, it could sidestep decades of engineering bottlenecks entirely. There's also a more philosophical layer to all of this that's hard to ignore. This research effectively demonstrates that evolution, entirely without understanding quantum mechanics, may have already been experimenting with structures capable of quantum behavior for millions of years. Fluorescent proteins evolve for reasons related to light, signaling, and biological function, long before any human scientist existed to study spin states or superposition. Now researchers are essentially asking evolution to keep innovating on their behalf. By carefully guiding processes like directed evolution in bacteria, adjusting selective pressures in controlled lab conditions, scientists can nudge these proteins toward becoming even better quantum sensors than nature ever needed them to be for its own purposes. That approach, called directed evolution, has already reshaped other fields of biotechnology. It's been used to engineer enzymes, design new proteins, and build biological tools that never existed in nature, until scientists steered evolution toward a specific human goal. Applying that same strategy to quantum sensing represents a genuinely new frontier, blending Darwin's oldest idea with one of physics' newest technologies. Let's zoom out for a second and think about why this story matters beyond the lab. Quantum computing has spent years being talked about as this distant futuristic technology, something that lives in research papers and tech company press releases, but rarely touches everyday life. This breakthrough starts closing that gap, not by making quantum computers more powerful, but by making quantum technology compatible with the actual conditions of life itself. Medical imaging is one obvious frontier, but it's far from the only one. Environmental scientists have discussed using biologically encoded quantum sensors to detect incredibly subtle chemical or magnetic changes in living organisms exposed to pollutants, toxins, or changing conditions, essentially giving researchers cellular-level early warning systems for environmental damage. Neuroscience is another area buzzing with potential. Brain activity involves incredibly small and fast electrical and magnetic signals. If protein-based quantum sensors could eventually be safely used to observe neurons at this scale, it could open new windows into understanding how thought, memory, and neurological disease actually work at a level current tools can't reach. Of course, none of this happens overnight. Every researcher involved in this work has been careful to frame it as an early but foundational step, not a finished product ready for hospitals or labs outside of specialized quantum research facilities. Photo bleaching still needs solving. Readout fidelity still needs improving. Scaling from a single protein qubit to reliable structured networks of them is still a major unsolved engineering challenge. But that's exactly how every transformative scientific breakthrough begins. The first working transistor wasn't a smartphone. The first sequenced genome wasn't personalized medicine. The first functioning qubit inside a living cell isn't going to immediately become a nanoscale MRI machine sitting in your local clinic. It's the first proof that the idea works at all, and history shows us that once an idea is proven possible, engineering tends to catch up faster than anyone expects. What makes this particular story stand out from typical quantum computing headlines is how it completely reframes what a quantum device even has to look like. For years, the public image of quantum computing has been dominated by giant, cold, expensive machines locked away in specialized government and corporate labs, completely disconnected from the biological world most of us actually live in and care about. This research suggests a very different possible future, one where quantum technology doesn't require isolating itself from life, but instead learns to work directly with it. Sensors built from the same biological material as the cells they're studying, quantum devices grown rather than manufactured. Precision achieved not despite the messiness of biology, but because of how that messiness has been refined over billions of years of evolution. It also raises a genuinely fascinating question that scientists are only beginning to seriously explore. If a relatively simple, naturally occurring fluorescent protein can function as a qubit under the right conditions, what other biological structures inside living organisms might already be capable of similar quantum behavior, structures nobody has thought to test yet, simply because nobody expected biology to survive quantum measurement in the first place. It's a question that naturally invites speculation, and it's important to separate genuine open scientific questions from overhyped claims. Some corners of the internet have already jumped straight to suggesting this proves things like quantum consciousness, or that the human body is secretly a giant quantum computer running your thoughts. That is not what this research shows. What it actually shows, carefully and specifically, is that one particular protein can function as a qubit under controlled experimental conditions. That's already remarkable enough on its own without needing to exaggerate it into something bigger than what the data actually supports. That question alone could shape an entire new area of scientific research for the next decade. Quantum biology, once considered a fringe or speculative idea by many mainstream physicists, is increasingly being taken seriously as a legitimate field, backed by peer-reviewed publications in journals like Nature, funded by major institutions like the National Science Foundation, and now anchored by dedicated research centers specifically built around it. The University of Chicago, through the Pritzker School of Molecular Engineering and the Chicago Quantum Exchange, has positioned itself directly at the center of that shift. Combined with initiatives like their collaboration with quantum computing, company IonQ, the university is clearly betting that the next major leap in quantum technology won't come purely from better refrigeration or better silicon chips, but from a genuinely interdisciplinary approach that treats biology as a partner in engineering, not just a subject to be studied separately. It also helps explain why Chicago, specifically, has become such a major hub for this kind of research. The city is now home to one of the largest concentrations of quantum research infrastructure in the country. bringing together university labs, national laboratories, and private industry partners all working on different pieces of the same broader puzzle. Having biologists, chemists, and quantum physicists working literally down the hall from one another turns out to matter a lot when your breakthrough depends on ideas colliding across fields that never used to talk to each other. Looking ahead toward the end of this decade, researchers involved in this space have talked about a realistic goal of pushing genetically encoded quantum sensors toward practical single-molecule sensitivity by around 2030. That timeline isn't a guarantee. Science rarely moves on a fixed schedule, but it gives us a rough sense of how soon some of these applications might start moving from research papers into real experimental tools used across biology and medicine. So where does that leave us? A team of researchers took a glowing jellyfish family protein, engineered it into a functioning qubit, and successfully detected real measurable quantum spin behavior inside living mammalian cells and bacteria at room temperature without any of the extreme isolation quantum systems have always required. That result alone earned recognition as one of the top scientific breakthroughs of the year, and it's already reshaping how top researchers think about the future of both quantum technology and medicine. It's not a finished quantum computer sitting inside your bloodstream, not yet. But it is proof that the century-old wall separating fragile quantum systems from the warm, noisy, chaotic environment of living biology isn't as solid as everyone once assumed. And once a wall like that cracks, even slightly, it tends not to stay standing for very long. Science tends to move this way more often than people expect. A single result, published quietly in a journal most people will never read, ends up rewriting the assumptions an entire field had accepted for generations. It doesn't arrive with fireworks. It arrives as a research paper, a handful of headlines, and a small community of specialists who immediately understand that the rules just changed. Everyone else usually catches up years later once the applications built on top of it start showing up in the real world. The next few years are going to be genuinely exciting to watch. Researchers are already working on improving stability, boosting sensitivity, and scaling from single protein qubits toward more complex, organized biological quantum systems. If they succeed, the line between biology and quantum technology, two fields that were treated as almost entirely separate for most of scientific history, may end up disappearing completely. And if that happens, the most powerful quantum sensors of the future might not be built in a lab at all. They might be grown inside living cells by biology itself, doing what it has always done best, adapting, evolving, and quietly solving problems long before we even knew how to ask the right questions. Think about how strange that idea really is when you sit with it for a moment. For 100 years, the entire quantum computing industry has been built around the assumption that fragile quantum states and messy biological life simply cannot coexist. Billions of dollars have gone into refrigeration systems, vacuum chambers, and vibration isolation, all in an effort to keep quantum information away from exactly the kind of warm, noisy, unpredictable environment your own body runs on every single second of every single day. And then a team of researchers in Chicago took a protein your cells already know how to make, shined a light on it, and found working quantum behavior sitting there the whole time. Not created from scratch through years of exotic materials science, but coaxed out of something biology had already been quietly building for millions of years, for entirely different reasons of its own. That's really the core of why this story deserves attention beyond just being another quantum computing headline. It's not simply a story about faster computers or more sensitive lab instruments. It's a story about how much we still don't know about the systems already running inside every living thing on this planet, including the one you're using to watch this video right now. If this kind of deep dive into cutting-edge science is exactly what you come to this channel for, make sure you're subscribed, because there is a lot more of this coming. Stories like this one, quiet breakthroughs that quietly rewrite what we thought was physically possible, don't usually get the attention they deserve, and that's exactly the gap this channel is here to fill.